All right. So my name is Jessica Stachler. I'm one of the co-founders of Phage Directory, and Jan Zhang is also here. He's my co-host. We started Phage Directory. We wanted to do PHAVES because we noticed there was a need. It's starting that everybody's jumping on this wagon of hosting virtual events, us included. But a few months ago, it just seemed kind of like a dark and lonely place, and there was nowhere to talk to each other, obviously. So we started it in response to the community pretty much asking us for it. And we're really excited to this time start a seminar version. So last time we had kind of an informal Q&A, then breakout rooms, and the breakout rooms were a big hit. The Q&A was super interactive. So this time we're doing a different format, and this is going to be our plan. We're going to shift formats, so you may be in for surprises. And Elyse Stachler from Eawag in Switzerland is our speaker today. So she's going to be giving about a 20 to 25-minute talk on her research. And then there's going to be a Q&A for her. I'll moderate that. And you can feel free to type your questions in the chat that you have all throughout her talk. We won't interrupt her unless there's something really, important that she needs to know, like, we cannot hear you. But if you put your questions in the chat, we'll get to them during the Q&A and we'll read them out just to keep it easier since there's so many of you here. So after that, we're aiming for the second half of the hour, probably a little over, to start breakout rooms. And we're going to do a 10-minute breakout room. And everyone's going to be randomized into rooms of about 4 of you. So you could just talk casually, introduce yourself. Where are you, how are you in the phage world? Are you a researcher? Maybe you want to talk about your research or what brought you here. It's just meant to have some friendship continue, and it's really not, don't be all stressed that it's an elevator pitch. I would just treat it like a casual chat, like after a conference, you're leaving the room and you might say hi to somebody. And hopefully you can get each other's information in that span of time if you do want to keep talking. And we'll come back to the lobby. I'll kind of bring us back here. After that, if there's more questions that came in for Elyse, we will answer some more of those then. And then we'll do, if we have time, another breakout room. So that is the kind of sequence of events. And yeah, if you have any technical issues, there's like annoying sounds, or you can't hear someone, or people need to mute, type them, and Jan is going to monitor the chat for technical issues. So I will get going then, and I'm going to introduce Elyse. So just quickly, we're spotlighting her now. This is Elyse Stachler. She's a postdoc at Eawag, which is Switzerland's National Water Research Institute. So Eawag is a fancy German acronym, and I like it. And so her research focuses on isolating phages from the environment and looking at how they interact with biofilms. And she is combining them and seeing how they work in combination with disinfectants, like chemical disinfectants. So that's what we get to hear about today, and I'm going to turn it over to Elyse now.
Great. So now I have my people in the corner that I can see. So first of all, I just want to thank Jessica and Jan and the people at Phage Directories for putting this kind of event together. It's honestly just really a great opportunity in order for us to share some research while conferences are not a thing right now. So I'm excited to get into my research looking at the effects of bacteriophage treatment with chemical disinfection on surface-associated Pseudomonas aeruginosa. But first, I just want to, so first I just want to introduce myself briefly. So I got my bachelor's and my doctorate in civil and environmental engineering from the University of Notre Dame and then the University of Pittsburgh. And my PhD thesis was centered on making a novel human fecal source tracking monitoring tool based off of Phage. So if you're familiar, maybe some of you have heard of Phage before. This is my Twitter handle up here. Just a shameless plug for you to follow me on Twitter if you're curious to hear what I have to say. And now I'm doing my postdoc in Switzerland, and it was really awesome. I was able to convince them to let me work on exactly what I want to work on, which brings me here today. So this is the point of the talk where I would introduce you all to phages, but since this is a phage-focused group of people, I'm going to assume that you all are already aware of the many advantages of phages and also the many challenges of using phages for treatment. So instead, we're going to do a poll, and Jessica, if you could put up the poll. So recently on Twitter, there was a poll going around about viruses, whether they were considered alive or not. And I'm just curious to see how the 41 people here, how we stack up with those people of Twitter. So I don't know if you all can see the live results as they come in, or maybe it's just me.
I don't know either, but I'm going to end the poll in about 10 seconds. So get your answers in.
Yeah, so the poll on Twitter, actually about 50% of people said it's complicated. Oh no, this was our test poll from yesterday. And about 25% each said living and not living, and we're pretty close to that actually, but it seems like we have more people who are wavering in our crowd. So we can go ahead and stop sharing the poll, I guess, if we can. Okay. So moving on, I just want to touch on opportunistic pathogens and specifically Pseudomonas originosa, since that's what I'm looking at studying. And many of these pathogens are common in our drinking water or source water, and they're often making up the biofilms in our drinking water pipes, etc. And they're not necessarily a problem, except when you get large amounts of these bacteria popping up. And particularly if they then come in contact with someone who is, technical difficulties, sorry, with someone who is immunocompromised. So these are susceptible populations such as the elderly or people with underlying health conditions. And Pseudomonas aeruginosa in and of itself is one of the escape pathogens, which you've maybe heard of before. And it's also considered a WHO priority pathogen. And Pseudomonas aeruginosa is particularly important in the hospital environment. It can easily cause hospital-acquired infections, and it particularly affects the lungs or the skin. So I really want to motivate my research by walking through an example. So here's a shower, and there's going to be Pseudomonas aeruginosa in our shower. Hopefully not this much, but you get the point. And so imagine we have an individual who has cystic fibrosis. These people are very much prone to Pseudomonas aeruginosa infection, and maybe they become so ill that they need to go to the hospital. And so there's many different sources of the bacteria in the hospital room itself. One is the patient, and then the patient can be coughing, which can produce foments, which collect on surfaces. And then as well, depending on what kind of treatment the patient needs, they may receive a catheter or an IV or a tracheal tube, and these soft plastics can often become colonized by biofilms. Now, all of these different areas are places where phage treatment could be used. For instance, on the patient itself, this is traditionally kept to compassionate care cases, at least in the Western world as of now. These biofilms and catheters or IVs, these can be treated or pre-treated with phages to eliminate these biofilms. And then as well, we could use sort of a disinfectant product that's made with phages to try and eliminate these other bacteria that are caused by foments in the room. But let's say in this case, our patient improves. And so some sources of the bacteria are eliminated, But there's going to be some bacteria that are lingering in the room between the patients, and this increases the risk of acquiring an infection in the hospital. Now, typically, these bacteria are eliminated through disinfection. This is our current best practice. But we know that these bacteria are often very resistant to traditional disinfectants. They can develop resistance, or they can be more naturally resistant if they're living within biofilms. So these can often stick around, and then if we have another patient that moves into that room, say for instance someone who has a burn wound, which can also be very likely to be infected by Pseudomonas aeruginosa, and you have a healthcare worker that comes over here, is interacting with different things and then interacting with the patient can become recolonized by this bacteria. And now this has created a hospital-acquired infection, an infection that the individual didn't have when they came into the hospital, but acquired within the hospital. And these hospital-acquired infections are quite a big deal. Pseudomonas aeruginosa alone is responsible for about 51,000 infections just in the US every year, and about 13% of those are multi-drug resistant. And in high-income countries, about 7% of all patients who go into a hospital acquire a secondary bacterial infection while they are within the hospital, and this is even higher in low-income countries at about 16%. And these secondary infections are often multi-drug resistant. And just to put this in perspective, probably a lot of you have heard this statistic before, but about 1 in 7 COVID-19 patients develop a secondary bacterial infection while they are admitted into the hospital, and as many as 50% of the deaths that are being attributed to COVID-19 have a secondary bacterial infection. So these hospital-acquired infections can really affect patient outcome. And so I've become very interested in sort of exploring the microbiome of the built environment, how we could potentially engineer that built environment microbiome in order to improve human health. So specifically, I want to go to my research questions. So I'm specifically looking at combining phage treatment with disinfection to see if this improves removal of bacteria. So my first question is, can combining these two treatments more effectively eliminate surface-associated Pseudomonas aeruginosa? The next question is, does the order of those treatments matter? I'm going to be applying my treatments sequentially. And then the third question is, how do the disinfectants affect the phages themselves? And the reason for this is sort of twofold. So I'm also very much interested in studying unintended consequences of widespread phage application. And I think that we should be a little bit careful before we implement something like this widely. And so one reason for applying disinfection after phage treatment is that we could eliminate active phage particles, so we don't have these being released into the environment. And then also as well, if phages are not susceptible to the disinfectant, then maybe these treatments could be applied simultaneously instead of sequentially. So getting into what I actually did. So I had 3 different scenarios here, and each one of these represent a well in a multi-well plate. So in this first scenario, I'm considering this my wet biofilm model. And here you put the media into a well, in this case in a 96 well plate, and you have the biofilm growing on the edge of the well at the air-water interface. Then when we move to this model, this is what I consider my dry biofilm model. It's exactly the same, except we have a plastic cover slip that is inserted perpendicularly to the bottom of the well. And this was done in a 12 well plate. And then you get a biofilm that forms on the cover slip, again, at the air-water interface. And you can remove this cover slip in order to wash the biofilm. And then we dried it for 2 hours before treating it. And then in the last scenario, it's very similar, except we have the cover slip lying flat in the bottom of the well, and we spot inoculate some of our bacterial host onto the cover slip, and then we dried it for 30 minutes before treating. So for the wet biofilms, actually in all of the scenarios, I used 2 different phages, JG4, which I just bought from a strain collection, and then phage P1, which I isolated from wastewater here in Zurich. And then the disinfectant I used for this study was sodium hypochlorite. And with the wet biofilms, I looked at treatment both phage followed by the chlorine, and then chlorine followed by the phage. And for all of these experiments, I analyzed the removal by flow cytometry, looking at the total cell count and the intact cell count. The dry biofilm experiments are exactly the same, except I only looked at phage treatment followed by disinfectant treatment. And then for the spot inoculations, again, only looked at phage treatment followed by disinfection. But here we included another disinfectant, benzalkonium chloride, which is a quaternary ammonium compound. That's a common surface disinfectant. But just for the sake of time today, I'm not going to talk about those results, and I'm just going to focus on the intact cell counts. So let's get to some data. So these graphs are for our phage JG4 for the wet biofilms. And on this side, we have phage treatment followed by chlorine treatment, and then this side, chlorine treatment followed by phage treatment. On these graphs, you see the phage concentration that was applied in increasing order. And on the y-axis, we have the Intelsat cell count, the log 10 reduction. And this is all normalized to the positive control. And then the different colors on the graph correspond to the different concentrations of chlorine that we tested against. So 0 ppm is black, green is 5 ppm, blue is 20 ppm, and red is 200 ppm. And so these graphs make a lot of sense. As you increase the phage concentration, you're getting increased removal of our host cells from the biofilm. And as well as chlorine concentration increases, we again see increased removal. But I really wanted to see if the combined treatment is better than either of the individual treatments alone. So this is how I decided to represent this. So basically each one of these cells indicates one of the combined treatments. And essentially the colors are telling you if it significantly varies from the chlorine treatment alone and the phage treatment alone. So essentially if you're seeing a blue color, it means that the chlorine treatment dominates. If you see a yellow color, it means the phage treatment dominates. And if you see a green color, it means that it's statistically significantly different from both individual treatments separately. So there's some sort of synergy here, but it's not necessarily even additive or more. So again, this makes a lot of sense. At low phage concentrations, we see that the treatment is dominated by the chlorine. At low chlorine concentrations, we see that the treatment is dominated by the phage. And again, we see the same thing here with the chlorine first treatment, except we see some evidence that chlorine first is not as useful. We don't get quite as good removal. And this is even more evident with our other phage, actually. So if we look here, this is the same data as I was showing on the other graph, but for our second phage. And if we look at the chlorine first treatment here, it's very much dominated by the chlorine treatment, except at our low chlorine concentration, when we get to really high phage numbers, then we do see some synergy. But this is really showing that it's not as useful. And my hypothesis for this is that when we treat with chlorine, we're stressing the cells, and the phages just aren't able to infect as efficiently anymore. So moving on to the dry biofilms. These are the same plots as I showed you in the previous slide. Here we have increasing concentrations of chlorine, of our phage P1 and phage JG4. But here, when we were looking at this, with increasing chlorine concentrations, we maybe get some removal, but it's not all that much. But really, when we treat with phage, we see no removal at all. And this was a bit surprising to us, but we hypothesized that maybe because we're stressing our cells, we're drying them, and we know that phages need active hosts in order to infect, that maybe this was determining the efficiency of the system. So in order to test this, and also I forgot to mention, I just want to give a special shout out to my master's student, Anina Cole. She did the work for the dry biofilms and the spot inoculations. So to test this hypothesis, we did some growth curves of our dry biofilms where we grew the biofilm overnight, and then we recovered the cells and then did growth curves with them. So the difference here is that these were dried for 0 hours and these were dried for 2 hours. And as you can see, there's no significant difference between the growth rate or the carrying capacity of the two communities. But there is a significant difference in the lag time in these communities. So for the 0 hours drying, it's about 3 hours, and for the 2 hours of drying, it increases to about 7 hours. And we were only treating our biofilms for about 4 hours with phages. So when we see this, this makes a lot of sense. So if we wanted to see removal in these dry biofilms, we would have to increase our time that we're treating with phages, maybe to 8 hours or even more. But we decided that this is not ideal. So this is why we decided to change over to doing the spot inoculation test. So again, here it's the same log 10 reduction graphs. When we look at sodium hypochlorite, again, with increasing concentration, we do see some removal. But then with our phages, even at very low concentrations, we see very large amounts of removal. And these dotted lines on the graph is our limit of detection on our flow cytometer. So even at 10 to the 3 PFU per milliliter, we're getting almost 3 log removal, or 10 to the 4 for JG4, because JG4 tends to be a little bit worse than P1 in removing cells. But Anina also did some combination experiments here, combining the disinfectants with the phages, but essentially because our phages do so well in this model, we had to use such low phage concentrations that the treatment is all dominated by chlorine. So I'm not showing those graphs here. And then finally, our last data is looking at how the disinfectant affects the phages in and of themselves. So here we have inactivation curves for JG4 and P1 at 5 ppm chlorine concentration. But essentially what you're seeing here is we have a drop off in phage concentration, and then it stays constant. And this is essentially because the phages are using up all the chlorine demand in the system, and that's why you get it tailed off like that. But what this does tell us is that these phages are susceptible to chlorine inactivation. And this is ongoing work to fully flesh out how these phages respond to the disinfectant. But if we look into the literature, we know that phages often vary in their susceptibility to different disinfectants. So this would really need to be monitored for each phage individually if you were going to use it. So if we go back to the research questions, can combining phage with disinfection more effectively eliminate surface-associated Pseudomonas aeruginosa? We do see some evidence that phage and disinfectant together can improve removal. Does the order of the treatments matter? It does seem that phage treatment followed by disinfection is more effective. And what is the effect of disinfectants on the phages? In our case, both phages are susceptible to chlorine. Still need to test this with benzalkonium chloride, but again, there's a wide variability in literature. So can we use the disinfectant to doubly eliminate the active phage particles? Maybe. And can we use the disinfectant and the phages together for treatment? Maybe. This is very dependent on the disinfectant and the phage. So looking into the future and what to do next, there's plenty of more research questions to be done here. But as 2020 has shown us, it can be kind of hard to plan for the future right now. So some of the experiments that I would like to do is looking at this system, but in more realistic conditions, where I treated these biofilms at their preferred temperature in nutrient-rich media. I would also really like to look at multi-species biofilms and how non-host bacteria react to phage treatment. And again, I'm also doing research into sort of some of the potential unintended consequences of phage treatment. But with that, I just want to thank a few people. I want to thank the Environmental Microbiology Department at Eawag for funding me through their postdoc fellowship. I also want to thank my specific group, Pathogens in Human Health, led by Tim Julian, although our group meetings look a bit different these days. And then again, I want to especially thank Amina Cole, my master's student who contributed to this research. And again, Phage Directory for giving me the platform to present my research. So with that, I am happy to take some of your questions.
Yay, awesome. Thank you, Elyse. Here's some clapping for you because it's hard to have this and everyone else is clapping. Okay. Yay, that was amazing. And yeah, I'm gonna have a look at the chat now because I saw some questions coming in. And I think we'll leave your slides sharing for now. And we'll keep this part on the video just to remind everybody. So we have a comment first. Wow, greater than 5% of hospital patients acquire secondary infections. Is that correct, Elyse? 5% of hospital patients?
Yes, so I would say that this is ICU patients.
Okay, yeah. And how did you isolate the phage from wastewater? That's from Kata.
Yeah, great. So it's very simple to isolate phages. Essentially, you gather the wastewater, you centrifuge or filter out all the other crap that's in it. And the theory is that if the phage is there, it will grow up. So there's kind of two different things. You can first isolate the host from the environment you're looking for, and if the host is there, then maybe the phage is there. In my case, I specifically seeded in Pseudomonas originosa strain PA01 because that's what I was interested in studying. And essentially you spike in your host, and if the phage is there, it will infect and grow up. And then you have, you just plate double agar layer overlay of this, you get an individual pack and you can re-isolate it a few times.
Awesome. By combining, this is from Fazal, by combining treatment of chlorine and phages, did chlorine not disrupt the phages? I think you did touch on this, but I'll let you.
Yeah, so essentially this is why I looked at doing sequential treatment. So either chlorine first followed by phage or phage followed by chlorine. Yeah, if you have more questions about that, feel free, but I also talked about the inactivation of the phages.
And I think that one came in kind of early in the talk anyway. So you did get to it. From Anna, do you sometimes observe enhanced biofilm formation upon phage treatment? In brackets, lysis leads to eDNA release, leads to more biofilm?
Yeah, so I did not, but that would be an interesting question. There certainly are some species that can build a stronger biofilm. We've seen this in the literature, but I did not witness this in my specific case.
Great. Okay, from Natalia, what is the difference between total and intact cells, and how do you measure them to target total/intact?
Yeah, great question. So with flow cytometry, essentially what you're doing is you have a stream of cells that you stain with a different dye, a fluorescent dye, and you can stream these cells in and then shoot a laser at it, and you see kind of how big the cell is. And the difference between total and intact is that you add propydium iodine, which can enter into a cell that is not fully intact. And then this quenches the dye. So essentially, if a total cell count, it includes all the dead and alive cells, because this dye just intercalates with the DNA of the bacterium. But then when you have the propydium iodine, this can also get into the cell if it's damaged, and then this eliminates the signal from that cell.
Very cool. We have a tip also, a suggestion to try two-way ANOVA if you haven't, with interactions for a statistical test of combined effects of phage and chlorine. Yes,
that is exactly what I did. I just don't have the interaction numbers here.
Got it.
Yes, thank you.
Yeah. Is it possible that phage will become resistant to chlorine after some time, from Atif?
Yeah, so this is a really interesting question. I would assume that they wouldn't, or it would be at a much lower rate than bacteria, because phage themselves need an active host in order to replicate. So they could maybe pick up resistance from their host if their host has some sort of genes that help them with this, but I think it would be not so likely.
Yeah, got it. We have some great talk, Elyse. Thank you, Elyse. I'm going to read those out too, because those are also important. And interesting work on the synergy of chemical and biological methods. Basic and very useful work. How did you check the stability of phages in sodium hypochlorite?
Yeah. So for this, I just took my phages in buffered solution and added a certain sodium hypochlorite concentration, and then you sample every so often, and of course adding sodium thiosulfate to quench the chlorine so that it doesn't continue to inactivate the phages.
Interesting. Have you ever looked, this is my question on a follow-on on that, but do you ever look under the microscope at what a phage looks like after you pour chlorine on it? Does it disintegrate it just like anything else?
I have not. I'm not a microscope person.
Yeah,
So.
I understand. Okay, we'll get to, these last three questions quick. So, and then we're going to go to the breakout. So based on your research till now, do you think phage can have a role as a hospital/clinic disinfectant, either independently or in combination with disinfectants?
So I think it's I mean, of course, this is a bit difficult of a question, but I think there's really potential there, especially because in these hospital environments, we're really so concerned with these antimicrobial resistant bacteria, and phages are really a way to get around this. And we have also seen a lot of evidence for even if a bacteria becomes resistant to the phage, then they become more susceptible to the antibiotic. So particularly in hospital settings, I think this really has a lot of usefulness. It's really hard to not be able to see the people to see if they're satisfied with the answers or not.
That's a good point. You can feel free to switch your screen to gallery because everyone knows that we're recording, so they accept. But we're almost at the end of questions anyway, so I think this is just a tip I'll share in the future for presenters. Okay, hitting these last couple questions, do you think phage encapsulation will improve outcome?
I'm not sure.
Cool, yeah. We've stumped her.
Hard to say. Yeah.